Hello everyone, now let us see the anatomy of urinary system, part 3. In the current session, we will be focusing on nephrons are the functional units of the kidney. Each nephron consists of two parts, a renal corpuscle where blood plasma is filtered and a renal tubule into which the filtered fluid or glomerular filtrate passes. The two components of the renal corpuscle are the glomerulus or the capillary network and the glomerular capsule or Bowman's capsule, a double-walled epithelial cup that surrounds the glomerular capillaries. Then the blood plasma is filtered. The blood plasma is filtered in the glomerular capsule and then the filtered fluid passes into the renal tubule, which has three main sections. In the order that fluid passes through them, the renal tubule consists of proximal convoluted tubule, nephron loop or loop of Henle, and distal convoluted tubule. The proximal denotes the part of the tubule attached to the glomerular capsule and distal denotes parts that is further away. And convoluted means tubule is tightly coiled rather than straight. The renal corpuscle and both convoluted tubules lie within the renal cortex, and the nephron loop extends into the renal medulla, makes a hairpin turn and then returns to the renal cortex. Here you can see this is the nephron, and the parts or components of nephron are renal corpuscle. This is the proximal convoluted tubule, and this is the distal convoluted tubule, and this is the nephron loop or loop of Henle. This is the descending limb of nephron loop and this is the ascending limb of nephron loop. This is renal cortex and this is the renal medulla. These are the renal papillae and these are the minor calyx. The distal convoluted tubules of several nephrons empty into a single collecting duct or CD. The collecting ducts then unite and converge into several hundred large papillary ducts which drain into the minor calyces. The collecting ducts and papillary ducts extend from the renal cortex through the renal medulla to the renal pelvis. So, one kidney has about 1 million nephrons, but as much as smaller number of collecting ducts and even fewer papillary ducts. In a nephron, the nephron loop connects the proximal and the distal convoluted tubules. Nephron loop connects the proximal and distal convoluted tubules. The first part of the nephron loop begins at the point where the proximal convoluted tubule takes its final turn downwards. And it begins in the renal cortex and extends downwards into the renal medulla where it is called as descending limb of nephron loop. It then makes a hairpin turn and returns to the renal cortex where it terminates at the distal convoluted tubule and is known as ascending limb of nephron loop. This is called as ascending limb of nephron loop. About 80 to 85 of the nephrons are cortical nephrons. The renal corpuscles lie in the outer portion of the renal cortex and they have short nephron loops that lie mainly in the cortex and penetrate only into the outer region of the renal medulla. The short nephron loops receive their blood supply from peritubular capillaries that arise from the efferent arterioles. The other 15 to 20% of the nephrons, 80 to 85% of the nephrons are cortical nephrons, and the other 15 to 20% of the nephrons are juxtamedullary nephrons. Juxta means near to, juxtamedullary nephrons. The renal corpuscles lie deep in the cortex, close to the medulla, and they have a long nephron loop that extends into the deepest region of the medulla. The long nephron loops receive their blood supply from the peritubular capillaries and from the vasa recta that arise from the efferent arterioles. In addition, the ascending limb of the nephron loop of juxtamedullary nephron consists of two portions. The ascending limb of the nephron loop of juxtamedullary nephron consists of two portions, thin ascending limb followed by thick ascending limb. The lumen of the thin ascending limb is same as the other areas of the renal tubule. It is only the epithelium that is thinner. The nephrons with long nephron loop enable the kidneys to excrete very dilute or very concentrate urine. The nephrons with long nephron loops enable the kidneys to excrete very dilute or very concentrated. Coming to the histology of nephrons and the collecting duct. A single layer of epithelial cells forms the entire wall of the glomerular capsule, renal tubule and ducts. However, each part has distinctive histological features that reflects its particular function. Let us see in the order of glomerular capsule, renal tubule and collecting duct. First of all, coming to glomerular capsule. The glomerular or Bowman's capsule consists of visceral and parietal layers. The visceral layer consists of modified simple squamous epithelial cells called as podocytes. And the many foot-like projections of these cells wrap around the single layer of endothelial cells of the glomerular capillaries and form the inner wall of the capsule. And the parietal layer of the glomerular capsule consists of simple squamous epithelium and forms the outer wall of the capsule. Visceral layer forms the inner wall of the capsule, whereas the parietal layer of the glomerular capsule, it consists of simple squamous epithelium and forms the outer wall of the capsule. The parietal layer of the glomerular capsule consists of simple squamous epithelium and forms the outer wall of the capsule. The fluid filtered from the glomerular capillaries enters the capsular space, that is the space between the two layers of the glomerular capsule, which is the lumen of the urinary tube. Coming to the histology of renal tubule and collecting duct. In each nephron, the final part of the ascending limb of the nephron loop makes contact with the afferent arteriole serving that renal corpuscle. Because the columnar tube cells in this region are crowded together, they are known as the macula densa. Alongside the macula densa, the wall of afferent arteriole and sometimes the efferent arteriole contains modified smooth muscle fibers called as juxtaglomerular cells or JD. Together with the macula densa, they constitute the juxtaglomerular apparatus. The juxtaglomerular apparatus helps regulate the blood pressure within the kidneys. The function of juxtaglomerular apparatus is to regulate the blood pressure within the kidneys. The distal convoluted tubule begins a short distance past the macula densa. In the last part of the distal convoluted tubule and continuing into the collecting ducts, two different types of cells are present. The most are principal cells, which have receptors for both antidiuretic hormone and aldosterone, the two hormones that regulate their functions. And a small number are intercalated cells which play a role in the homeostasis of blood pH. The collecting ducts drain into large papillary ducts which are lined by simple columnar epithelium. Generally, the number of the nephrons is usually constant from the birth, and any increase in the size is solely due to the growth of individual nephrons. If the nephrons are injured and become deceased, new ones do not form. And signs of kidney dysfunction usually do not become apparent until the function declines to less than 25% of normal, because remaining functional nephrons adapt to handle a larger than normal load. And the surgical removal of one kidney, for example, stimulates the hypertrophy or enlargement of the remaining kidney, which eventually is able to filter blood at 80% of the rate of two normal kidneys. Coming to the overview of renal physiology, to produce urine, nephrons and the collecting ducts perform three basic processes. They are glomerular filtration, tubular reabsorption, and tubular secretion. First of all, coming to glomerular filtration. In the first step of urine production, water and most solutes in the blood plasma move across the walls of the glomerular capillaries, where they are filtered and move into the glomerular capsule and then into the renal tubule. The next step is tubular reabsorption. As filtered fluid flows through the renal tubules and through the collecting ducts, the tubule cells reabsorb about 99% of the filtered water and many useful solutes. The water and solutes return to the blood and it flows through the peritubular capillaries and vasa recta. Note that the term reabsorption refers to the return of substances to the bloodstream. Reabsorption is nothing but return of substances to the bloodstream. And the term absorption by contrast means entry of new substances into the blood as it occurs in the gastrointestinal tract. And the final step is tubular secretion. A filtered fluid flows through the renal tubules and collecting ducts, and the renal tubule and the collecting ducts secrete other materials such as waste, drugs, and excisions into the fluid. Note that the tubular secretion removes a substance from the blood. Tubular secretion removes substance from the blood. Solutes and the fluid that drain into the minor and major calyces and renal pelvis constitute the urine and are excreted. And the rate of urinary excretion of any solute is equal to its rate of glomerular filtration plus rate of secretion minus rate of reabsorption. And by filtering, reabsorbing and secreting, nephrons help maintain homeostasis of blood volume and its composition. Here you can see the schematic representation of renal physiology. This is an efferent arteriole and this is an efferent arteriole. Efferent arterioles enters and efferent arteriole it leaves. And this is the glomerular capsule. This part is the renal corpuscle and this part is the renal tubule and collecting duct. So first step is glomerular filtration. In this, the glomerulus, blood plasma and dissolved substances smaller than most proteins get filtered into the glomerular capsule. And here, tubular reabsorption takes place. This is the step where tubular reabsorption takes place. Along the renal tubule and collecting duct, water, ions and other substances get reabsorbed from the renal tubule lumen into the peritubular capillaries. From the renal tubule into the peritubular capillaries, they get reabsorbed. Hence the term tubular reabsorption. And ultimately they enter the blood. And the third step is tubular secretion. All along the renal tubule and collecting duct, substances such as waste, drugs and excess ions get secreted from the peritubular capillaries. Tubular secretion means drug substances, waste and other excess ion. They get secreted from this peritubular capillaries into the renal tubule. This is the term tubular secretion. And these substances are ultimately excreted through urine. And this is the final passageway for urine which contains the secreted substances. And here is the blood, it contains the reabsorbed substances. So second step is tubular reabsorption. In tubular reabsorption, majority of the water, ions and other substances, they get reabsorbed from the renal tubule into the peritubular capillaries. From the renal tubule, here the absorption takes place into the peritubular capillaries, whereas tubular secretion, here the solutes are secreted from the peritubular capillaries into the renal tubule. So the three steps are, the first step is glomerular filtration, the next is tubular reabsorption and finally tubular secretion. Thank you for watching. Please subscribe for further videos on medical coding and CPC training.